I'd like to thank the organizers for this opportunity to present some of our work. We've developed the assay for evaluating serological levels of antibodies against COVID-19 and measuring the immune response. And we've done this with a number of populations, including immunocompromised populations. So I would like to, again, thank them for this opportunity to present this work. The COVID-19 pandemic doesn't really require any introduction at this point, but it still rages on despite the introduction of a vaccine at a record pace and has impacted over 243 million people worldwide. Similarly, it has induced the death of these across the world at nearly 5 million people worldwide. This has emerged as one of the hottest topics in research science as well as medicine, and one that we have engaged. But what's interesting is the lack of diagnostic measures reported early on in the pandemic has led to a need for these type assays to be developed. But what's interesting is that has evolved at a rapid pace over the course of the pandemic. As the disease progressed and as new innovations came online, such as the COVID-19 vaccine, the needs that the medical community had in order to better serve the populations has also evolved. And so this started off initially with, incidents, trying to document in the population what portion of that has actually seen a COVID-19 infection. And what's interesting is that there's mild or asymptomatic patients, which were going undocumented for the most part. The numbers that I previously presented were confirmed cases, and those are typically symptomatic and most commonly those that have severe symptoms. And so the ability to really understand the impact of this disease overall and its incidence is something that was of great interest early on. But this element really petered out as soon as the vaccine was introduced late last year. With that, once the vaccine was available, then it became more of like a question of who's responding to this vaccine, how strong is their response, how durable is that immune response, and both in normal populations as well as those that are immunocompromised. This sort of question induces further evolution. As everyone knows, a booster shot program has been implemented in the United States. And so when is the optimum time to give that? The CDC and the FDA agree that 6 months is a reasonable time frame, but I'll show you some data which suggests that maybe sooner might be more appropriate. And then finally, there's the idea of like breakthrough infections, being able to identify those. So with this, what we mean is for those individuals that have been vaccinated against COVID-19 that go on to acquire COVID-19, so become infected, what is the rationale for that? Is it because they have diminished immunity, that is, the vaccine is worn off, as it were, or are there variants that are able to bypass the vaccination program? So these are the sorts of questions that we sought to answer or start to address with this undertaking. We started off by developing an assay. And the assay that we wanted to develop, so this is back in February of last year, we developed a multiplex assay built on the Luminex platform that would evaluate, that is, measure titers for multiple antigens present with the SARS-CoV-2 virus. The spike S1 domain, or alternatively, sometimes we use the spike RBD domain, which will be more important with the vaccine patients, was the first one that we sought to develop. And then with that, primarily because of its role in infection, that's the primary molecule involved in how the virus infects cells. But also there's the nucleocapsid protein and the matrix proteins, as well as the envelope proteins, which are readily immunogenic and produce signals that can be readily ascertained through an assay, a serological assay like we developed. And we wanted an assay that produced quantitative results that could be monitored over the course of time. This assay, we wanted to evaluate both neutralizing and non-neutralizing titers to give an appreciation of who's been infected and what their level of protection is as a result of that infection. And then we also wanted to be able to evaluate multiple types of immunoglobulins. And so that is, being able to watch the seroconversion. So the maturation of the immune response with time. Most of the efforts that I'll be showing you up front focus on the immunoglobulin G titers. And that's primarily from the FDA's edict that any approved tests would need to monitor exclusively IgGs. But this assay, and this is inherent to Luminex assays and this design of assay, that we require a very small amount of samples. Less than 2 microliters is required for our assays. And I'll talk more about that in a second. First, I wanted to go into a little bit of the Luminex platform for those that may or may not be familiar with that. It's basically a bead-based ELISA type format. I'm not going to present schematics here for sandwich assays, which are probably what most people are familiar with. This is alternatively, this is an indirect assay, which means our analyte is the antibody that binds to an antigen. The antigen for the ELISA is bound to a plate prior to it is one of the first steps in the assay. Then is exposed to the patient's sample, which has the antibodies in it. The antibodies bind to the appropriate antigen. And then there's a detection system, which is usually, in this case, an anti-human antibody. That anti-human antibody is labeled and that produces your signal. In case of most ELISAs, that's HRP or alkaline phosphatase. But in Luminex, it's a little bit different, where now we're using a fluorescent signal, but it's the same system as far as like how the assay is designed. But in lieu of a single antigen being bound to a plate, we bind that antigen onto a bead. This microsphere has a coating, which the Luminex instrument, which we're using the xMAP 3D in this case, can distinguish up to 500 different types of assays simultaneously. So with that, our 4-assay design is able to be done concurrently in the same well. So that saves a lot of time and money as far as processing patient specimens. And then, so we developed the initial draft of the assay, and this included 4 different analytes. And so it's the spike S1, the nucleocapsid, the membrane, and the envelope glycoproteins. The envelope glycoprotein later fell out of the assay, so we ended up with a 3-plex, primarily from the standpoint of what it was adding to our performance was negligible. In our validation studies, where we tested over 1,000 patients, we had, again, initially this is for seroprevalence, that we used over 1,000 negative patients. And for the most part, these were a median age that was on the upper end of the scale, because at that time, we were most worried about the elderly population. And then we also had a cohort from that were confirmed COVID-19 positive by PCR. And these cases were, they're 44 in number versus the 1,000. And we're able to evaluate these and look at our performance and how we're able to distinguish who were positive for COVID-19 previously. Pardon me. What we found was we had a perfect, a near perfect sensitivity and specificity. And so we had a sensitivity of 100%, which means we correctly classified all of the COVID positive cases. Of the COVID negative cases, we only had 4 misclassifications. Those misclassifications likely resulted from an infection from a non-COVID-19 coronavirus. But that's a very low percent, and that's similar to what other assays were showing at the time, necessary for FDA approval. And so our overall accuracy was over 99%, and that's again in over 1,000 patients. And so with that, we had an assay that we felt pretty confident in that could be used for doing research, and also had applicability for guiding clinical management of patients in the future. A feature of this that I didn't point out earlier is that this workflow is also fully automated, and just with a single instrument, we're capable of processing over 1,000 patient specimens per day with automation. And so with that, we have an assay that we feel pretty confident with and has high performance and is high throughput. And just as a side note, this assay is also highly amenable to using microsampling techniques. We're a big fan of dried blood spots. And so a lot of the evaluations that we performed in some of the early studies and currently are performed in the dried blood spot format, as opposed to requiring phlebotomy to be performed. This allowed us to mail collection kits to patients and instruct them over the phone on how to collect just 4 drops of blood go into this window here, and then we collect the resulting serum/plasma from the window up here. And with that, we have a sample that has almost perfect analytical concordance. That is, you get the same answer whether you're using serum or plasma across these populations. So that's a very convenient way to get specimens in the future. And so just to kind of recap that very quickly. So we developed an assay. We're happy with the performance of the assay. It had over 99% accuracy. And with that, we're measuring 3 different antigens from the SARS-CoV-2 virus, which allowed us to really have good classification efficiency and good confidence in our results. Our results required at least 2 positives of the 3 to return a positive result. And so we're able to greatly reduce our false positives that we produced, which is, of course, critical for that aspect. We also, which I'll show you some data on this in the later slides, we paired this assay with a pseudovirus neutralization assay, so we can look to see if our results that we're measuring of antibodies corresponds with a functional ability for those antibodies to neutralize. I'll go through that in just a moment. But back to the slide for a second. So that work that we just presented was designed to impact a study looking at incidence. That is, trying to decide or determine what portion of the population has been affected by COVID-19. The next question that really came up had to do with the vaccine. So introduction of the vaccine in December of last year really kind of changed the game. Now we had a means to potentially prevent or minimize the impact of this pandemic. And we wanted to really evaluate like the normal populations, but also contrast those with immunocompromised populations. Those that received organ transplant, had cancer, etc., are ones that we're actively evaluating now, and trying to determine when, do they get the same immune response as the normal populations, and if not, to what degree are they protected from infections? Also of interest is the durability of the immune response, and trying to determine, how long are you protected if you've been vaccinated? And then that kind of leads us to, the booster shot program, where we really want to understand when's the optimum time to give that. And so with boosters, this is current numbers from the World Health Organization. That has really had a very substantial impact on the course of the disease. While numbers are still growing and the disease is still raging, its true impact is muted somewhat by the fact that good portions of the populations, depending on where you're at, have been vaccinated at this time. Trying to evaluate those is that immune response and how durable that is, as I mentioned, something we're very interested in. And so with that, we took our assay that I briefly described the development, and we wanted to look at, that is, quantify the immune response to the vaccine. And for this, we're primarily using the RBD assay at first, but later we ended up using the S1 quite a bit. The S1 assay is a larger antigen, and it includes the RBD peptide, and it allows us to evaluate neutralizing peptides beyond what the vaccine provides as a primary response. The nucleocapsid and the membrane assays were included to allow us to evaluate previous COVID-19 infections, and also has application for identifying breakthrough cases. So cases that were negative serologically for having an infection going into a vaccination. After that, we only observe an RBD signal or an S1 signal, so a spike signal. If they develop nucleocapsid or membrane titers after that, that means they're a breakthrough case and they've contracted COVID-19 after vaccination. And we wanted to do this as well with the immunocompromised populations. But more importantly, we wanted to be able to monitor seroconversion in these populations. We wanted to be able to not only measure the IgG, so the mature immune response, but look at the time course of how the immune response at different phases with different immunoglobulins was progressing. So with that, we took advantage of some innovations in the instrumentation from Luminex in order to really focus in on this question. And the INTELLIFLEX instrument was introduced earlier this year, and we had one of the beta test models that we performed some of these studies. And what's interesting about this is, versus this instrument is, whereas before I described the Luminex platform as having the coded beads, which would allow us to distinguish and run the three assays in unison, that's shown on the left here, and use a phycoerythrin secondary antibody as a reporting system. So this allowed us to evaluate an IgG alone or an IgA alone, or along those lines. With the INTELLIFLEX, we're actually able to evaluate two of these in unison. And so we can do IgG with IgM, for example, in the same well, and evaluating the identical specimen. This greatly improved our throughput in doing these evaluations, saved us a lot of money as far as like running these assays, as well as time. That greatly improved our throughput as well. And that was only available on the INTELLIFLEX. The ability to measure multiple epitopes in the same well for the identical analyte is something that is, for the most part, limited to just a couple different technologies. And with the introduction of this instrument, that expanded that out to the Luminex platform. And so with that, the goal of this project was to monitor a timeline of the seroconversion from patients that are becoming vaccinated and concurrently evaluate multiple immunoglobulins in the same assay, and using our 3-plex COVID-19 panel at the same, in doing this. So again, this is the spike S1, the nucleocapsid, and the membrane glycoprotein. The membrane glycoprotein, by the way, is also known as the matrix glycoprotein, depending on what circles you are part of. So I'll go through a little bit of the assay development for this and show you how we optimize this assay to run with this dual reporter type system, and how we got that ready to start these sorts of evaluations. So the first thing that I want to show you is that we use Analytical Standards for the immunoglobulin G type assays. And for those, these follow a typical sigmoidal type curve. You can see that most dramatically here with the nucleocapsid. This is showing 3 distinct batches, and the reproducibility of these is very high. And it follows a nice S shape, so it's a sigmoidal type shape. These other ones, which are not showing the plateau phase, that occurs at higher concentrations, just we're not showing that as part of this particular curves. The relevant concentrations that we need to evaluate these are in the range that's shown here. So these ranges represent what we need to evaluate human patient specimens. We didn't need to go higher, so that's why those aren't represented. So one of the first elements that we really wanted to look at in assay performance was precision. So we looked at two elements of this, both intra-assay precision as well as inter-assay precision. This slide I'm showing currently is showing intra-assay precision, and this is within the same batch. If we ran that multiple times, how reproducible were the results? The cornerstone for this is anything under 25% is considered acceptable, and as you can see from our results, a majority of these were well below 25%. We did have one that started to approach 25%, but that was still acceptable. Looking at inter-assay precision with this slide, this is looking at multiple batches of the beads as we produced them in our laboratory. And again, the cornerstone for this is 25%, and all of the points that we evaluated were again under 25%. Some of them didn't again get into double digits, but given that these were an acceptable level, we thought it acceptable to move on with the assay. So the precision of this assay was considered acceptable. Next, we get into some incubation time optimizations. The first, of course, being the evaluation of the specimen capture time. That is, how long do you incubate your sample with your assay? And we did this up to 4 hours for each of the trials that we did. And again, we're looking at the spike S1, the membrane, and the nucleocapsid assays. And we did this looking at all three serotypes, so IgM, G, and A. The 4-hour time point was the normalization point, so we made that the 100% of our one in this case. And what we found was at 4 hours, you captured 100% of your sample, but you really get into more of a economy of time for performing the assay with this sort of analysis. That is, you have a higher throughput if you perform the assay for half the time, for example. You can double your throughput for the most part. And so with that, most of the assays that we performed did a capture that was in the 120-minute time frame. But some of these, if we're just looking at IgG, we're able to actually go down to 30 minutes alone. And that's something that we did pretty routinely for our results. And so with that, we now have an appreciation of the incubation time for the specimen. After that, we went into looking at the secondary antibody incubation times. Here we're looking at how long for our anti-human antibody, which has our reporter directly conjugated to it, either phycoerythrin, in this case for IgA, or the super bright 436 for IgM, how long do those incubations need to be? And we saw a pretty flat result for most of these, indicating that by 2 hours, we're at the 100% or the normalization point, but as low as 60 minutes or even 30 minutes, as we are seeing here, we had still reasonable saturation of our signal. And so with that, 30-minute incubation times for the secondaries were selected. And the nucleocapsid proteins showed similar sorts of trends, where for the IgA-IgM combination for the dual reporter system, that for the most part, the 2-hour time point gave the highest value, but the other time points we tested gave similar values, a little bit lower, yes, but the reproducibility of these were pretty comparable, meaning that is highly reproducible, whether you did it at 30 minutes or 120 minutes, just a little bit of a difference in the magnitude of the signal that we measured. With that, we were able to determine that we could go to a 30-minute time point for some of these incubations and still have quantifiable results. In the next experiment, we're really examining here the idea of having bleed over from one channel to the other. So we wanted to make sure the results that we obtained were going to be robust. And with that, we ran sets of experiments where we had standards in one channel, and the other channel we left blank. And the idea here that can be seen much better in this next slide is the idea that the blank series, which here is indicated in red, versus the series that has a signal, there's a very low percentage of the signal that bleeds over. And this is seen consistently throughout the different traces. And this indicates that the bleed over in the INTELLIFLEX system is very small. And so we don't have to really worry about bleed over, fluorescent bleed over in these assays. So when we're quantifying the results, we feel pretty confident in the values that we're measuring as far as quantitation. There's some more examples of those. Again, really contrasting the one versus the other. And for all these, it's a very low percentage of the total system. And so with that, the evaluations that we perform looking at multiple serotypes is pretty robust. After that, so now we're getting into the actual patient specimens now. So here's where we're looking at the longitudinal. This is looking at multiple patients at different time points. This is pre-vaccination over here. So this is like at the day of vaccination before they get it. So this is their first shot here. The changes that occur, and then with the second dotted line, that's their second vaccination for the primary program. And what you see, so there's immunoglobulin G, M, and A represented here. Again, these were done in unison, and IgG, IgM, dual reporter was done for all three analytes, and then the IgA, IgM was done as well. And these were all COVID naive patients, which means there was no signal for the the nucleocapsid or the membrane assays. What you actually see here is there's a pretty quick immune response. So the IgG is at maximum values by like 15 days. For the IgM, that comes up in a similar time frame. It's not super dramatic beforehand, as you might expect for other immune responses. And for IgA, what's interesting here is that came up in a similar manner, but dropped down really fast, versus the IgM persisted to some extent over the course of that time, only going down slightly over the 120-day period for these being watched. But the IgG is really persistent. And IgG is the one that we look at as being the true measure of the immune response. And also, please take care to note the magnitude of the scale differences here. The levels that we're seeing with the IgG were in a magnitude of 30,000 versus 800 for IgM. So the IgG response was quite dramatic relative to the IgM and IgA. These same patients, when we looked at them at a longer time point, so what I just showed you was out to 3 months, so it's out to here. If you look further down the road, so out to 8 months, this signal continues to degrade. And so the red here is the titers that we're measuring for RBD in this case. And the 6-month time point is where the FDA approved the booster shots to be distributed. But as you see, based on the maximum titers that we observe up here, that at 6 months you are down to about 15 to 20% of the original maximum values. The amount of protection there was in question. Because of that, we decided to run neutralization assays along with these. And so with neutralization, so that's in green here, that the higher the number, the lower the neutralizing potential. That is, the less effective the antibodies are. So down here, we don't have a very high titer, and so it takes a lot of the neutralization is very low. That is, it has high numbers. But as the peak happens for the IgG, in this case, those numbers come way down. That is, the neutralization potential increased dramatically. And as these titers decay, so the neutralization also increases. So with that, we're seeing a good concordance between the ability to measure titers of the RBD antibodies, and the ability for those to actually neutralize. And so that makes sense, that if you have the antibody presence, you're able to neutralize. And then finally, in looking at some of these populations, the black here represent normal populations, and the blue and the red represent immunocompromised populations. And with that, we see not only a lower initial concentration of the spike protein. Now, there's a lot of heterogeneity within these populations, but as a general statement, the concentrations of the titers for the neutralizing antibodies, that is the spike-based antibodies, in immunocompromised individuals is lower than those of normal populations. There's a lot of interest right now with the booster program to see if that's in fact changed. There's several reports out currently which seem to indicate that with the third dose that immunocompromised individuals might actually get similar benefits from that as quote unquote normal populations. But there's a definite lower initial immune response to vaccination in immunocompromised individuals. And so that really brought us to this third question here, which I introduced. And that's the source of our current efforts, is focusing on booster shot administration for immunocompromised populations. That is, does this third booster, the third shot of the vaccine, provide protection which is comparable to those which normal populations are receiving? And initial indications is yes, it looks very positive. How durable that is will be a topic that we'll be monitoring very closely and hopefully have some findings on in the near future. And so just to wrap up, we developed an assay that is able to very rapidly measure titers from the major antigens associated with the SARS-CoV-2 virus. We can measure multiple isotypes of the immune response, that is different serotypes, so IgAs, IgMs, and IgGs using the INTELLIFLEX system. And this is a very cost-effective way to monitor the immune response as well as document immunity in populations. And again, based on that one set of data I just showed you, that by 2 to 3 months, populations have about 50% of their original immune response, and that response is near baseline by 6 months, which is when booster shots are currently recommended. So perhaps those, with greater populations, that's something that maybe something that we can submit to the CDC or the FDA as a means to see if that can be moved up in time for certain individuals. And thank you at this point. And the team that was responsible for the work I just presented was Imad Tarhoni, the postdoc in my lab, and Christina Fayed. David Gerard was very integral to these studies as well. And at this point, I'd be happy to take any questions.